Chapter 3, the cell. There are two basic types of cells, prokaryotic and eukaryotic. Prokaryotic cells include the bacteria and archaea. They're structurally simpler, typically smaller, and they lack membrane-bound organelles. This is an image of a bacterial cell. You can see where it contains a cell wall that we do not have as eukaryotes. They have a plasma membrane. Bacteria also have DNA, but it's not located within a nucleus. It's free-floating in the cytoplasm. But they do have ribosomes, which are the organelles in which proteins are produced. Bacterial cells have different proteins than found in eukaryotes, and this is why antibiotics work, is that they target the prokaryotic protein and cellular components that are not found in the eukaryotic or in the human cell. Eukaryotic cells are typically larger and structurally more complex. They do have membrane-bound organelles. Eukaryotic cells include plants, animals, fungi, and protists. Organelle simply just means little organs. Organelles are components within the cells that perform a specific function. There are more than 10 trillion different cells specializing in more than 100 different functions in the human body. For example, neurons in the brain coordinate perception and intellectual functioning. Kidney cells continuously remove waste and reabsorb components returned to the bloodstream that the body is going to maintain. Muscle cells contract in response to electrical signals from the brain. You may refer to this image in your textbook. These are some of the organelles or the components that we will be discussing within the chapter. This image just gives a good breakdown of each of the different organelles within the eukaryotic cell. This table also breaks down the prokaryotic and eukaryotic. You can see one of the primary differences between the prokaryotic and eukaryotic is in membrane-bound organelles are absent in prokaryotic, present in eukaryotic. The DNA is going to be located in the nucleus in eukaryotes, where it's just in the cytoplasm in prokaryotic. Cells vary in size, but they can never exceed the volume that could be nourished by materials passing through the surface membrane. The surface to volume ratio takes into account that as a cell gets larger, its surface area increases much more slowly than its volume. Therefore, a large cell would have difficulty moving all the nutrients it needs and all the waste it produces across an inadequate surface, and thus would die. Most eukaryotic and prokaryotic cells are typically measured in micrometers and can be viewed either by a light or electron microscope. This is an image of a striated muscle cell with the light microscope. Striated cells B with the transmission electron microscope, you can see it in much greater detail, and a striated muscle with a scanning electron microscope. So these are just different viewings of the same striated muscle cell with three different types of microscopes. Although we begin life as only one cell, that cell differentiates into many specialized cells. And these specialized cells have structures that reflect their particular function. In this image, a sperm is specialized to be highly mobile, in contrast to the egg, which is large and immobile. The egg is going to be packed with material needed to initiate development and support that embryo until the placenta is set and formed. A mature red blood cell is devoid of most organelles, is specialized for carrying oxygen. It has the protein hemoglobin. A cardiac muscle cell is specialized for contraction. It contains numerous gap junctions called intercalated discs that allow for the propagation of the signal for contraction from cardiac cell to cardiac cell. The plasma membrane is the outer boundary of the eukaryotic cell, and it controls the movement of substances into and out of the cell. The primary components of all plasma membranes are phospholipids. The phospholipid bilayer separates the extracellular fluid from the material inside the cell contained in the cytoplasm. So the phospholipid bilayer separates the extracellular from the intracellular fluids. This plasma membrane of phospholipids, they have a hydrophilic head facing outwardly. Hydrophilic means water-loving. So extracellularly and intracellularly, these hydrophilic heads, and they have hydrophobic tails from each of the bilayers that faces inward. Proteins, cholesterol, and carbohydrates are also part of the membrane and give it the qualities of a fluid mosaic. The proteins are interspersed among the lipid molecules like tiles in a mosaic. Many proteins are able to move sideways to a slight degree through the bilayer, giving the membrane its fluid quality. So this shows the plasma membrane with the phospholipid bilayer. You see the extracellular, and inside the cytoplasm, that would be the intracellular fluid. So the yellow components are the hydrophilic heads with the hydrophobic tails facing inward from both layers. You see an embedded protein with a carbohydrate. You see the glycoprotein. Glyco just means it's a carbohydrate attached to a protein. The glycolipid is a carbohydrate attached to a lipid. These are all types of surface markers that will interact within the body, that act as receptors. Functions of the plasma membrane is that, number one, it maintains the structural integrity of the cell. Number two, it regulates movement of substances into and out of the cell. 3, it provides recognition between cells, such as the glycoproteins that we looked on the previous image. Glycoproteins means a carbohydrate attached to a protein. Glycolipids, as we reviewed on the previous screen, are membrane carbohydrates that are attached to phospholipids that function as recognition signals for cell interactions. Plasma membranes also provide communication between cells. They contain these receptors, these proteins, including glycoproteins, glycolipids, embedded in the cell plasma membrane. And they also contain, they stick cells together to form tissues and organs with CAMs. These are called cell adhesion molecules. These cell adhesion molecules are particularly important in developing embryos. Cancer cells lack contact inhibition, thus they form tumors. Movement across the plasma membrane, it can be passive or active transport. Passive transport is movement across the membrane that does not require energy. It's passive. Passive transport includes simple diffusion, facilitated diffusion, and osmosis. Active transport, on the other hand, is movement across the membrane that requires energy. So active is going to require energy in the form of ATP. Passive does not require energy. Simple diffusion is just random movement of a substance from a region of higher concentration to a region of lower concentration. The end result is just an equal distribution of the two substances, of the substances in the two areas, so it eliminates the concentration gradient. So simple diffusion is just the movement of molecules from an area of higher concentration to an area of lower concentration, eliminating that concentration gradient and equalizing distribution of the substances in the two areas. It does not require any energy. This talks about a lipid-soluble substance that moves through the lipid bilayer from high to low concentration. A lipid-soluble substance may be something like a steroid hormone, and because of the lipid substance and the plasma membrane also being a lipid substance, it is able to move through the plasma membrane from the extracellular fluids, such as a steroid hormone that may be carried into the blood to diffuse into the cytoplasm, to be carried to the nucleus. So it's going to go from an area of high concentration to an area of low concentration with no input of energy. Facilitated diffusion is the movement of a substance from a region of higher concentration to a region of lower concentration with the aid of a membrane protein. Thus, it's called facilitated. Water-soluble substances need to be assisted or facilitated by certain proteins called carrier proteins to cross a cell. So facilitated diffusion again is going to go from an area of high to low concentration, but it is going to require the aid of a carrier protein, thus it's facilitated, but again, because it's diffusion, it does not require energy. This shows an image of a glucose moving through the lipid bilayer from high to low concentration with the aid of a carrier protein, facilitated diffusion. So it's going to come up to the plasma membrane. You see carrier protein opening up. The glucose molecule is going to bind. It's going to be transported to the inside of the cytoplasm, where this glucose will be used to produce ATP cellular respiration during cellular respiration. Osmosis is the movement of water across a selectively permeable membrane, across a phospholipid membrane, from a region of higher water concentration to a region of lower water concentration until equilibrium is reached. Osmosis and diffusion are basically the same process, except osmosis deals when we're talking with water. It does not require energy. It's going to go from low to high. So it's the movement of water in osmosis. This image talks about the hypo, and isotonic solution. We are basically an isotonic environment. So hypertonic just means that you have a higher solute concentration and less water concentration. A hypotonic solution means that you have a lower solute concentration, thus you would have a higher water concentration. So in a hypertonic, the solute concentration is higher than the water concentration. So if a cell were put into a hypertonic, more water would move out of the cell and thus it would shrink. In a hypotonic solution, the solute concentration is lower than the water concentration. An isotonic cell placed in a hypotonic solution, the water would move therefore into the cell and it would swell and burst. Isotonics are the same solute and water concentration, thus there would be no net movement in either direction and the cell maintains its original shape.